So we'll start with our metabolism review. The 3 parts of metabolism. The majority is going to be in this first part. We'll do carbs, go through the whole pathway of the carbohydrates, just talk about how lipids, where they step in, and then what's going on with proteins. So glucose processing, who can tell me some terms for glucose processing? There's glucogenesis, it's making glucose. Then what's the term for breaking down glucose? Glycogenolysis. You're breaking down, or breaking down glycogen. Sorry, I termed that wrong. Let me go here. Yeah, glycogenesis is forming glycogen. Glycogenolysis is breaking down glycogen. So one is sending glycogen, forming glycogen in the form of temporary storage of energy. And then when you need more glucose later, then you're going to go through glycogenolysis to bring your glucose levels up. Gluconeogenesis happens when you create glucose through non-carb sources. What would be a byproduct of gluconeogenesis? Ketones. Excellent. So glycolysis. Where is glycolysis going to take place in a cell? Not the mitochondria, so that would put it out in the cytosol. Destiny's on it. So then, what does it produce? What do we get out of glycolysis? Get 2 ATP, 2 pyruvic acids, and 2 NADHs. Yep. So what happens to pyruvic acid in an aerobic state? Pyruvic acid turns into what? Acetyl-CoA. Maybe I should, I'll write that down. So we go pyruvic acid is going to become acetyl-CoA. Acetyl-CoA is going to go where? It's going to go into, I'm going to do a little dotted line. A little here, because we're now in the mitochondria. Okay, so it's going to go into the Krebs cycle. The Krebs cycle creates what? It is going to create, yes, carbon dioxide. That's sort of its waste product that we get from the Krebs cycle. And what else does it make? We get a couple of ATP that come out. That's kind of handy. We get then more that we're going to move along will be these more NADHs, FADH2. So we're going to leave that be since we're here at this carbohydrate. So as far as glycolysis goes, we got 2 ATP, we got 2 NADHs, which we'll also send over here and talk about it in a moment. Then we get the pyruvic acid. Pyruvic acid becomes acetyl-CoA so it can enter the mitochondria and enter this Krebs cycle. During anaerobic conditions, which really means we made a bunch of pyruvic acid, we turn it into acetyl-CoA, and we run it through the Krebs cycle, and we use oxygen later through the electron transport chain. This is still going, but shoving more acetyl-CoA, it can't handle anymore. It's just using it and burning it as quickly as it can. But our body needs even more energy. So we continue anaerobically by doing more glycolysis so we can get the 2 ATP out of glycolysis, but we're still making pyruvic acid, which is coming into acetyl-CoA and it backlogs. So, or because it's backlogging in its conversion to acetyl-CoA. So what does it become temporarily? Lactic acid. So then pyruvic acid, if it can't become acetyl-CoA, will come out and become lactic acid temporarily until the the movement of acetyl-CoA into the Krebs cycle is now sufficient to be able to take this lactic acid and turn it into acetyl-CoA, and then we can eventually use it for energy. So the Krebs cycle, where it takes place, the byproducts, and the end results. So where it takes place is in the mitochondria. A byproduct would be carbon dioxide. And the end results is we get some ATP, we have the CO2, but we're getting NADH, FADH2. These guys are like tokens for more ATP. So they are tokens where the NADH is going to become 3 ATP. The FADH2 is going to become 2 ATP. And this process is via the electron transport chain. In order to utilize the electron transport chain and make these ATP, we're going to need oxygen. So O2 is required for this reaction. So we don't just get 3 ATP and 2 ATP total. It's for every NADH we get 3 ATP, and we get many of them. So we get, like, was it 10 NADHs, and you can add them up. And anyway, so you get lots of them. That's just what they ultimately become. So you'd multiply it by however many you get. There we go. We covered all those things. Any questions? Okay, we'll go to lipids. So you should know the types and structures of lipids. So lipids could be a steroid. It could be a triglyceride. We spend more time talking about the triglycerides because that's the main one we use for energy. But the steroids are cholesterol-based, and they're used to make hormones. It's also considered a lipid. Then we also have phospholipids, which are used to make our cell membranes. So these are things that our body inside actually uses fat for a purpose, for a good, not just for energy, like triglycerides. So how we process lipid processing, and we'll talk about the triglycerides, is if we have a triglyceride, we have this glycerol backbone, and then again with the fatty acid chains, 3 of them. We digested them as triglycerides, broke it apart, absorbed the individual components, and reformulated them back together for storage in our adipose tissue. When we want to undergo lipolysis or lipolysis, we can break these bonds here, and then this glycerol backbone is actually going to go through glycolysis and end up being pyruvic acid. The fatty acid chains, they go through what's known as beta oxidation to break these long chains, which are carbon-linked chains, into carbon to carbon. So just a 2-carbon unit, which then can become acetyl-CoA, and we burn it through the Krebs cycle and the electron transport chain and redeem our ATP that way. So the energy that we get from the breakdown of triglycerides is enormous because of the long chains. If you think of a single triglyceride, how long each of these chains are. So just one of these carbon-to-carbon units makes almost as much as a single molecule of glucose, outside of the 2 ATP that glucose made from the cytosol. So there you can see how fat is an incredibly wonderful storage of energy and very, very helpful for people to transport their energy in times of uncertainty in terms of their food availability. What is ketogenesis? Formation of ketone bodies because you don't have carbohydrates to turn directly into glucose. The brain will only live off of glucose. So the body has to do some trickery and say, all right, I'm going to take the fat, turn it into glucose, but in doing so, ketones is this nasty byproduct that comes from that process. So it allows us, it's an adaptation that allows us to stay alive and keep our brain going, even if we're not eating carbohydrates. It's not a very efficient way to do things, but that is indeed a way that we can to help keep our brain survive. So ketogenesis is formation of ketones. in the absence of carbohydrates because we have to make glucose from a non-carbohydrate source, which would be gluconeogenesis. Last section is going to be protein metabolism. This is pretty simple in that we digest proteins. You should know about digestion of proteins in the stomach and the intestines. I threw in the brush border enzymes because there are some enzymes that just cleave the 2 amino acids. It also does that actually for carbohydrates, breaks the last 2 little saccharides into little monosaccharides. But the main thing for proteins with regard to metabolism and energy is the process is known as deamination. You pop off an amine group. That doesn't really mean anything to you guys until you take chemistry. So it really is taking this protein molecule, and if you pop off the amine group, it's called deamination obviously, then the protein can come in, and once it's deaminated, will become acetyl-CoA, and we can then burn it. So protein can be a very useful form of energy if we're starving to death and we have no other food coming in. Obviously, protein is not a primary source of energy our body wants because it really means that we're breaking down something that we would otherwise use for a good thing in building up our body. So the main thing is deamination. You should know that there were 20, are now 21 amino acids, different types of amino acids. We did talk about essential, non-essential, and conditional, but you don't need to know that for the point of this test. It's just, I pointed them out. So you should know that there are 20, have been 20, currently 21 different types of amino acids. And if we're going to utilize an amino acid, or sorry, going to utilize protein for energy, it must be deaminated. But the main thing, whether you're using fats or carbs or proteins, is they're all going to become acetyl-CoA. That then funnels into the mitochondria for the Krebs cycle, which makes carbon dioxide. The tokens that we get, NADH, FADH2, go through the electron transport chain, which is where we utilize oxygen, and this is where we get the bulk of our ATP that we live off of. And that's your metabolism story. Any questions?